Let's see, next on our agenda is we will have Magdalena Amézquita from Arizona State University College of Health Solutions. And we're going to talk about sleep patterns in early infancy associated with gut microbiota development.
Hello, my name is Magdalena, and I'm here to talk about sleep patterns in early infancy and how they are associated with gut microbiota development. Obesity worldwide has nearly tripled in the last 50 years, and many factors contribute to it, such as low poor food quality and increasing stress, but especially our environment. Our environment nowadays promotes low physical activity and less exposure to the outdoors. And this in turn gives you less exposure to natural sunlight and also less exposure to the microbes that live outside and can't enter through your air conditioner filter. Obesity prevalence in children and adolescents in the US was 19.3% in 2018. This was even before the pandemic. And Hispanics and people with low socioeconomic status represent the highest proportion of this population. Now, how does this relate to sleep? Sleep early life behaviors have been associated also with obesity, with rapid gain weight that in turn is associated with obesity, such as daytime nap quantity or the balance between nocturnal and daytime sleep. Now, even though we're just at the tip of the iceberg discovering how the gut microbiome influences our health, we now know that intestinal microbes have clear differences in comparing obese and lean individuals, and these may contribute to gain weight. And although we can change our gut microbiome later in life, this is pretty resilient and just to temporal changes. So this highlights how important it is the development during the early stages of life. Now, the gut microbiome has also circadian rhythmicity. Thais and collaborators found that about 10% of the gut microbiome fluctuates diurnally. And these disruptions in the circadian rhythm have been hypothesized as stressors and may have an influence on the gut microbiome. In the bottom, I borrowed two graphs from a sleep review where you can see in the left how in a mice that has a normal clock gene function, the firmicutes population is higher during the nighttime and lower during the daytime. And in mice that have lost that cycling, they're completely flat. So the analysis I'm presenting here today was done from using data from a randomized control trial where they tested how an education intervention could prevent the incidence of infant overweight. So pregnant Latina women with obesity were recruited, and their infants were measured at 1, 6, and 12 months of age. And they were measured by asking a brief infant sleep questionnaire, their weight, their height, and their eating patterns. So the goal was to prevent the obesity in here. And later on, they were followed up, and a subsample of 35 babies was used to collect their feces to measure their gut microbiome. So the samples were sent to ASU, and the DNA was extracted and amplified and sequenced. And later on, I performed a statistical analysis using QIIME2 and the Silva gene database to compare the diversity between the samples. Now, just as a brief reminder that I like to give myself for this, when we're looking at alpha diversity, I exemplified its diversity within the samples. So here the bouquet full of only tulips would have a low alpha diversity since it only has one species, but in the one to the right, it has two more, so it would have a higher alpha diversity. And beta diversity compares differences between samples. So in this case, the bouquets have completely different flowers, so they would have a higher difference in their community structure. Now, the objective of this study was to measure how these sleep behaviors impact the gut microbiome composition at 3 years of age. So we compared total sleep time, bedtime, number of wakings per night, the total sleep time during the day, during the night, and we turned these into categorical variables to compare the gut microbiome based on them. We found that, for example, in this case, the gut diversity at 3 years was different depending on the bedtime the toddlers had. 6-month-old, that the infants had. The 6-month-old babies that slept earlier than 9 p.m. had a richer and more even microbiome composition when they were 3 years old than the babies that had later bedtimes. Here you can see the graph at the left, it's the Shannon index. So we're comparing how rich the gut microbiome was from those babies. And to the right, we see how even they were. So how the number of features per taxa that were within each sample. So in this case, earlier bedtimes, they were more even. We also found that the nighttime sleep proportion of the bedtimes affected the gut microbiome. In this case, it affected the richness and also phylogenetic diversity. Babies that obtained more of their sleep at night as a proportion of the total sleep time at 6 months old had a lower richness and lower phylogenetic diversity than the babies that slept less of their total sleep time during the nighttime. And as you can see, if they slept less during the nighttime, they might have slept more during the daytime. So we also saw that an increase in the nap times of the babies had also an influence on the gut microbiome and diversity. So babies that slept more than 5 hours as naps during the daytime had a higher alpha diversity than babies that slept less than 5 hours. And this was observed also in richness and phylogenetic diversity. Now, from these two samples, we also observed differences between the community structures. We performed 4 analyses to compare the sample communities, where we saw if microbes were present or absent, or the abundance which they had within in the samples, and how close or far they were within the phylogenetic tree. And we found that they were, with Permanova analysis, that they were statistically significant differences between the community structures. Here I exemplified between filled circles are babies that slept less than 5 hours during the daytime, and empty circles are babies that slept more than 5 hours during the daytime, and babies that slept more during the nighttime are in blue, and babies that slept less during the nighttime are in pink. And although it wasn't significant, babies that woke up more than twice at night when they were 6 months old had a lower richness of their alpha diversity. And it makes sense. So with the longer stretches, they had the higher diversity of their gut microbiome. So this, as a conclusion, these sleep patterns in early infancy do affect the gut microbiome when babies are gone into toddlerhood. And at 6 months of age, earlier bedtimes and the higher proportion of daytime sleep in infancy was associated with a more diverse gut. And we still need to observe how these associations are maintained in infants as they consolidate their sleep towards the nighttime. And more further work is needed on that, and to see if there's particular microbial biomarkers that influence these sleep behaviors, and to see how these changes affect health later in childhood. I'd like to thank my co-PIs, Dr. Whisner and Dr. Petrov, that them, along with Dr. Reifsnider and Dr. McCormick, did the parent study of the snuggle bug where we worked on right now. Thank you very much.
All right, thank you. That was really interesting. I see that there is a question in the chat. What factors were controlled for in the study? Breastfeeding, household stress, cortisol levels, socioeconomic factors, etc. Sorry if you said it and I missed it.
Hi everybody. So the study it was an interventional study with the education, and many factors like feeding were also observed. And my co-PI already published a paper where they associated the sleep factors with rapid gain weight. And we're also trying to study how they were associated with feeding patterns and sleep. So there were, I don't have them in hand right now, but many factors were controlled for, especially with feeding and sleeping.
Great. And maybe we can share that paper after.
Yeah. And also educational levels and like socioeconomic indexes were also addressed, and they're in that paper. I can share a link if you want.
Oh, great. I had a question too. I wondered if it's possible that the reverse relationship is also true, where the gut microbiome might have an impact on sleep. And so maybe kids with less diverse microbiome have less good sleep quality. Is that possible too?
Yes, definitely. And there's also many studies that have been done that observe like not a unidirectional way, but also like definitely the gut can impact sleep. And there's like different interventions that are being studied, like probiotic supplementation, but like most of those studies have been done in adults. So our Snugglebug project is one of the first that are studying it right at the beginning where everything is formed.
Very cool. Thank you.